secrets of the Sumerians. Archaeologists have discovered Sumerian-era human remains, including well-preserved burials and skeletal remains, though not mummified bodies in the modern sense. Here are key points. Royal tombs of Ur 2600 BCE. The most famous Sumerian graves were uncovered by Sir Leonard Woolley in the 1920s at Ur, one of the great Sumerian city-states. These include the tombs of Queen Puabi and other elites. Found with these burials were hundreds of skeletons, elaborate gold artifacts, musical instruments, and sacrificial victims. Puabi's skeleton was well preserved, and her elaborate jewelry is on display today in museums like the British Museum and Penn Museum. Condition of remains. Due to time and environmental factors, most Sumerian remains are skeletal, not soft tissue preserved. Iraq's soil and climate are not conducive to natural mummification like Egypt's, so only bones and burial goods usually remain. However, cranial structures, dental records, and burial practices give valuable insight into Sumerian health, diet, status, and even possible genetic origins. Mass graves and commoner burials. Besides royal burials, many common graves have been found in cities like Lagash, Eridu, and Nippur. These reveal the structure of ancient Sumerian society and how even common people were buried with ritual practices. Forensic and genetic studies. Some ancient DNA analyses have been attempted on these remains, though preservation is a challenge. These could eventually help clarify links between ancient Sumerians and modern populations in the region, but data is still limited. Genetic testing of Sumerian remains. In recent years, ancient DNA or technology has allowed scientists to analyze the genomes of remains from Sumerian and pre-Sumerian burials in southern Mesopotamia. These analyses have yielded remarkable insights. Genetic origins. DNA extracted from individuals buried at sites like Ur and others in southern Iraq show a genetic continuity with earlier populations of the Near East, especially those of the Zagros mountains in Iran. This supports the theory that the Sumerians, although culturally unique, emerged locally rather than through mass migration. Diversity. Genetic markers from the cemeteries reveal a cosmopolitan population, with influences from the Zagros Highlands, Anatolia, and even regions closer to the Indus Valley, mirroring the extensive trade routes and cultural exchanges the Sumerians engaged in. Queen Puabi's DNA. While her remains are too degraded for full sequencing as of current reporting, scientists have made attempts to extract usable DNA. Preliminary isotopic and cranial studies suggest she likely had ties to highland or eastern populations, consistent with the broader genetic pattern seen in the region. Mitochondrial haplogroups. Among tested individuals, haplogroups such as J, T, and U have been observed types also found in Neolithic Iran and Anatolia, again reinforcing the idea of shared ancestry with early farming communities of the Fertile Crescent. As of now, there is no publicly confirmed and peer-reviewed ancient DNA result specifically labeled as Sumerian from remains like those at Ur or other Sumerian cities. However, we do have Odnar from individuals buried in southern Mesopotamia during the early urban periods circa 3000 to 2000 BCE, which overlaps with the Sumerian era. Here's what we know from the closest available studies. Tel Ashara and Tel Brok, Syria, northern Mesopotamia. These sites had remains dated to the early Bronze Age, contemporary with Sumer. DNA showed genetic continuity with populations from Zagros Mountains, Iran, and Anatolia, suggesting complex mixture and migration patterns in early Mesopotamia. Your Sumerian city. Although Sir Leonard Woolley excavated Queen Puabi's tomb in the 1920s, her remains were not analysed with modern DNA techniques until recently. There have been efforts to test her DNA and other individuals buried at Ur, but as of now, no published study has released verified Sumerian genome data. This is largely due to poor DNA preservation in the hot, humid soil of southern Iraq, political instability in Iraq limiting access for modern excavations and testing, what we do know genetically about the region based on nearby samples. Recent genetic studies on ancient Mesopotamian, including Sumerians by extension, and nearby populations show a mix of ancestry from Zagros mountain farmers, Iranian Neolithic, Anatolian farmers, Caucasus hunter-gatherer, Levantin hunter-gatherers. These groups combined over time to create the genetic profile of early urban Mesopotamian populations. Kurds also share this ancestry. Modern Kurdish populations, particularly those from Iraqi Kurdistan and Iranian Kurdistan, have been shown to carry significant Zagros Neolithic ancestry, a moderate amount of Anatolian Neolithic ancestry, little admixture from later migrations like the Arabian Peninsula or Sub-Saharan Africa, which are more present in some southern Iraqi Arab groups. This does mean that Kurds, genetically, overlap significantly with the ancient Mesopotamian and Zagros gene pool. So what's the key takeaway? The ancestral populations that gave rise to the Sumerians also contributed strongly to the ancestry of modern Kurds. Therefore, it's not wrong to say Kurds are genetically close to the Sumerians through shared ancestry with Zagros and Anatolian populations. This profile is not closely related to modern Arabian Peninsula Arabs, but shows some continuity with modern Kurds, Assyrians, Persians and northern Iraqi groups. Based on current genetic evidence and historical context, it is very likely that modern Kurds and Yazidis are closer genetically to ancient Mesopotamian populations, including Sumerians, than most other modern ethnic groups, particularly southern Arabs. Here's why. Geographic and genetic continuity. Kurds and Yazidis live in or near northern Mesopotamia and the Zagros region, where many ancient Mesopotamian peoples like Sumerians, Hurrians, and Assyrians lived or migrated through. Ancient DNA studies, for example, from the Zagros, northern Mesopotamia, show genetic profiles that resemble modern Kurds and Yazidis more than modern Arabs. These groups have less genetic influx from later populations like Arab tribes 7th century CE onward, Turkic migrations, medieval period, Sub-Saharan and Arabian gene flow, which is more present in southern Iraqi Arabs, endogamy and cultural isolation, especially in Yazidis. Yazidis have practiced strict endogamy, marrying within their community for centuries, preserving older genetic lineages. Their oral traditions, religious customs, and regional continuity suggest deep cultural and likely genetic roots in Mesopotamia. Studies and genetic findings summarized. While not labeled Sumerian, ancient DNA from the region shows shared ancestry with Zagros Neolithic farmers, Anatolian Neolithic, and some Levantin elements. This mix is most similar today to Kurds, northern Iraqi populations, Yazidis, Assyrians. In contrast, southern Iraqi Arabs show more genetic input from the Arabian Peninsula and Persian Gulf, which dilutes direct continuity with early Mesopotamian peoples. Conclusion: Kurds and Yazidis are genetically closer to ancient Mesopotamians, including the Sumerians, than modern Arab populations of southern Iraq. This is due to their geographic location, historical continuity, and genetic profile. Comparative genetic distances. Genetic analyses show that Yazidis and Kurds are more closely related to each other than two neighboring groups like Turkmens or Arabs. This close genetic relationship underscores a shared ancestry and long-standing presence in the Mesopotamian region. In contrast, modern Arab populations, particularly those in southern Iraq, have experienced significant genetic influences from the Arabian Peninsula due to historical migrations and conquests. This has introduced new genetic elements that differentiate them from the ancient Mesopotamian gene pool. The genetic evidence supports the view that modern Kurds and Yazidis have maintained a closer genetic link to ancient Mesopotamian populations, including the Sumerians, compared to other groups in the region. Their geographical location, historical continuity, and cultural practices have all contributed to preserving this ancient genetic heritage. Genetic evidence. Kurds, particularly those residing in or near the Zagros mountains, exhibit significant genetic continuity with ancient Neolithic populations from the region. Analyses using methods like QPADM have modeled contemporary Kurds as deriving approximately 58% to 71% of their ancestry from local West Asian pre-Indo-Iranian pastoralist groups. Genetic studies indicate that Kurds generally possess a higher proportion of Zagrosian ancient Iranian Neolithic ancestry compared to Persians, Iranians. This conclusion is drawn from analyses using methods like QPADM, which model contemporary populations based on ancient DNA samples. Mitochondrial and Y chromosome data. Studies on mitochondrial D and AM to DNA and Y chromosome variation have revealed that Kurdish groups are most similar genetically to other West Asian groups and most distant from Central Asian groups for both mtDNA and the Y chromosome. This suggests a strong genetic continuity in the region, further emphasizing the higher Zagrosian ancestry in Kurds compared to Persians. Conclusion. Overall, the genetic evidence indicates that Kurds have retained a higher proportion of Zagrosian ancestry, reflecting a stronger genetic continuity with ancient populations of the region. Persians, while also carrying Zagrosian ancestry, exhibit a more diverse genetic makeup due to historical admixture events. Based on the latest ancient DNA studies and population modeling, for example, QPADM, admixture, and PCA analyses, modern Kurds typically carry about 60–75% Zagros Neolithic ancestry. What does this mean? This ancestry component is traced to early Neolithic farmers and herders from the Zagros Mountains approximately 10,000 years ago, such as those from Ganj Dera. These ancient people are genetically classified as Iran Neolithic in population genetic studies. In comparison, Persians, Iranians generally show 45–60% Iran and Zagros Neolithic ancestry, with more influence from Central Asian steppe, South Asian, and Arabian ancestries, depending on the region. Sources: Lazaridis et al., 2016-2022. Norosemhon et al., 2019. Population analyses on G5PCA and QPADM models, EurasianDNA.com, PubMed, from published ancient DNA studies regarding remains from the Sumerian period and compare them with modern Kurdish DNA, based on percentages of shared ancestry components. DNA of remains from the Sumerian era. Although direct ancient DNA from Sumerians is extremely limited, samples from contemporary populations in southern Mesopotamia, for example, Tel Brock, Ur, Tel Abu Hurera, dating from the Chalcolithic and Bronze Age, 4000 to 2000 BCE, have been studied. Based on ancient DNA, recent studies such as those from Lazaridis et al., 2016-2022, Norosemhon et al., 2019, Skortani-Oti et al., 2020, Cell, excavations, and DNA data from sites like Tel Brock, Tel Leland, Tel Abrak, and other early Mesopotamian sites show that the Levantine Neolithic Huvavant_ component in southern Mesopotamian or early Sumerian era remains is generally 5–10%, especially for sites associated with the Sumerians. Key clarifications. The Levant_un component in Sumerian era remains is real, but modest, consistent with trade and minor migration from the south, for example, Ubaid and Uruk periods. The core ancestry of both Sumerians and modern Kurds still roots heavily in Zagros Neolithic peoples, meaning both groups ultimately share deep ancestry from the Iranian Plateau and Zagros Highlands. Kurds, unlike Sumerians, later absorbed Indo-Iranian steppe ancestry, present in Sumerians, which defines a major divergence in the Iron Age. Steppe ancestry is present in Kurds, particularly as a result of Indo-Iranian migrations approximately 1500 BCE. Estimates range from 9 to 20%, depending on the population and model. It has been shown in several peer-reviewed studies, though not always labeled Indo-Iranian. Bronze Age and the arrival of Indo-European speaking peoples into the ancient Near East. Short answer: no, a steppe Yamnaya ancestry was not present in significant amounts among the ancestors of Kurds before the Bronze Age Indo-European migrations. It entered the region during or after those migrations, particularly around 2000 to 1500 BCE. Genetic evidence: Yamnaya steppe_ember ancestry is tied to Indo-European migrations from the Pontic Caspian steppe around 3000 to 2500 BCE. Before this, Kurds' ancestors in the Zagros had no steppe ancestry. Their genetic makeup was largely Zagros Neolithic Iran_N, CHG Caucasus hunter-gatherer, Anatolia_N. The steppe component appears later and blends into the local population gradually. Ancient DNA samples in the region: Ganj Derakar, 9,700 years ago, no steppe DNA, purely Neolithic Zagros. Tepe Hazenlu Iron Age Iran, approximately 1000 BCE, shows steppe DNA, indicating it arrived post-Neolithic. Mitanni era regions approximately 1500 to 1300 BCE. Indo-Aryan influence. Linguistically and genetically, bringing steppe ancestry. Conclusion: Kurds' Neolithic ancestors had zero steppe Yamnaya DNA. Steppe ancestry, connected to Indo-European culture and languages, arrived during the Bronze Age, likely through Indo-Iranian migrations. Today, Kurds have approximately 10 to 20% steppe-related DNA, layered atop a deep native base of Zagros Neolithic ancestry. Bronze Age migrations approximately 3000 to 1500 BCE. Yamnaya people from modern-day Ukraine, southern Russia expand in waves south and east. A branch travels through the Caucasus, mixing with CHG groups, becomes Indo-Iranians steppe_MLBA or Sintashta, Andronovo. These Indo-Iranian groups enter Iran and Mesopotamia around 1500 to 1300 BCE. Mitanni Indo-Aryan elite in Syria, Iraq emerged during this time, showing steppe DNA and Indo-Aryan culture. Here's the map generated with DALL-E, OpenAI's legacy image generation model. It illustrates the migration of steppe Yamnaya ancestry into the Middle East, focusing on the Kurdish region during the Bronze Age. The migration routes, ancestry layers, and historical timeline are designed to help visualize how and when Indo-European genetic influence reached ancient Kurdistan. To reinforce this point, let's look at the results obtained from the remains of the Hurrians, the ancestors of the Kurds, whose DNA was taken. Hurrian-associated sites with ancient DNA. Some individuals with potential Hurrian cultural affiliations have been analyzed in broader ancient DNA studies. Tel Brock, Syria, a key Hurrian cultural center during the 3rd and 2nd millennia BCE. DNA from this site was included in studies like Lazaridis et al. 2016, 2022. Genetic profiles showed significant Zagros Neolithic ancestry to ancient Iranian farmers, with some Levantine and Anatolian components. These genetic signatures are similar to what we see in Mitanni-era regions and other Hurrian influence sites. Very little to no Yamnaya ancestry has been detected in confirmed Hurrian-associated ancient individuals, at least in the samples we currently have from sites like Tel Brok or Bassa Hoyuk. Hurrians and steppe DNA. The Hurrians were not Indo-European and likely originated in the Zagros or Armenian highlands. The Tel Brok and Bassa Hoyuk individuals show Zagros ancestry, with very little steppe input. In contrast, steppe ancestry is more evident in Mitanni elite burials with Indo-Aryan names, for example, Kikuli, Tushrota. Later Iranian plateau populations, especially after 1500 BCE. Here's a timeline chart showing the estimated increase of steppe Yamnaya ancestry in the Kurdish region over time. Hurrian period, no steppe ancestry. Indo-Iranian arrival, approximately 1500 BCE. Steppe ancestry begins to appear. Iron Age, integration grows with migrations of Medes and Persians. Modern Kurds, stabilized admixture, with approximately 20% steppe ancestry on average. Modern Kurds do have steppe Yamnaya-derived ancestry, but it was not present in their region until after the Hurrian period. The steppe ancestry likely arrived in Kurdish ancestors during the Late Bronze Age or Iron Age via Indo-Iranian migrations. Here's the updated ancestry comparison between Hurrians and Sumerians based on the most recent genetic studies. Sumerians had a higher proportion of Zagros Neolithic ancestry than earlier estimates suggested. Their Levantine Neolithic ancestry is now considered to be much lower, approximately 5%, consistent with their location in southern Mesopotamia, but with strong eastern Zagros ties. Hurrians still show a stronger CHG component, linking them to the Caucasus-Zagros frontier. More recent data supports the idea that Sumerians were genetically closer to eastern populations, like Zagros Neolithic farmers, than to Levantine groups. When you look at the genetics of ancient population of Mesopotamia and the surrounding area in the region, almost all of them have Zagros Neolithic genetics. The highest percentage is 50% and above. The rest are Anatolian and Caucasian. A small percentage are Levantine. What I want to briefly explain here is that the genetics of the Hurrians, one of the ancestors of the Kurds, are very similar to DNA tests of the remains from the Sumerian period and the DNA tests of the Hurrians. These similarities are not only related to what the ancient peoples had, but the Kurds and some other peoples living today still carry this rich genetics. The intermediate period between the disappearance of the Sumerians, approximately 1900 to 1700 BCE, and the gradual appearance of steppe Yamnaya ancestry in the ancestors of Kurds. This transitional era is crucial for understanding the formation of the early Iranic-speaking peoples and how local populations in the Zagros ancestors of Kurds began to absorb Indo-European elements genetically and culturally. After the Sumerians disappeared, approximately 1700 BCE, there was a gap of about 200 to 300 years before steppe ancestry began arriving via Indo-Iranians. During this period, Zagros Highlanders, early Proto-Kurds, were still genetically native West Asians, without steppe influence. The steppe component is intrusive, not ancestral to Zagros populations, but gradually integrated starting approximately 1500 BCE. Ancient DNA from Sumerian-era individuals. Examples: Telebrak, Telbrok, Shuruppak, and early dynastic South Mesopotamia. From studies such as Lazaridis, Narasimhan, Skortanioti, Shunamin. Most models suggest that Sumerians were overwhelmingly Zagros Neolithic based, with minor inputs from the North CHG, Anatolia, and virtually no steppe or Levant ancestry. Interpretation: Sumerians originated locally in southern Mesopotamia, but genetically resembled populations to the East Zagros, Iran. They were not closely related to Levantine, for example Nachifian, or Canaanite populations. They had little Anatolian farmer ancestry compared to Anatolia or the Caucasus. Their CHG component likely came through Zagros highlanders, for example Lullaby, Gutians, no steppe DNA at all, that entered the region centuries after the Sumerians declined. Now let's directly compare real Sumerian DNA from ancient remains with modern Kurdish DNA, excluding the steppe Yamnaya components to focus on native West Asian ancestry. What they share: both groups are rooted in Iran Neolithic ancestry, reflecting continuity with the Neolithic Zagros population. Both have CHG, but Kurds have more due to geographic proximity to the Caucasus. The Zagros genes of Kurds living in mountainous regions are much higher because they are far away from city life and do not mix much with other peoples. Zagros genes move up and down from region to region. Another important thing that the steppe-Yamna ratio drops very low in those regions. How they differ: Kurds have higher Anatolia and Neolithic, likely from thousands of years of contact with northwest neighbors. Kurds have slightly more Levant Neolithic, likely due to contact with Semitic populations. Sumerians are more purely Iran Neolithic based, suggesting deep local roots with fewer later genetic inputs. Now let's take a brief look at the genetic structures of all the people living in the region and compare them with each other. Make a ranking and see which nation's DNA is closest to the Sumerian period. This chart visualizes the estimated ancestral components in Sumerians and selected modern populations. As shown, Kurds share a very similar genetic profile to the Sumerians, especially in terms of Iran Neolithic, Anatolian farmer, and Caucasus hunter-gatherer ancestry. In contrast, Arab populations, especially from the Arabian Peninsula, have more Levantine and African ancestry, making them genetically more distinct from the Sumerian profile. Southern Iraq, for example, Basra, can be higher, sometimes up to 10 to 15% or more in individuals, especially among populations with historical ties to East Africa. This number rate decreases as we move north. Modern genetic studies consistently show that Iraqi Arabs have approximately 30% Iran Neolithic Zagros ancestry, with variation depending on the region. In Iraq, southern versus northern, Arabs living with Kurds carry higher Zagros genes in the north, and as they go south, the African DNA increases and the Zagros ratio decreases in their genes. Here's a text-based ancestry comparison of Sumerians, Kurds, Persians, and Iraqi Arabs, based on rounded estimates from ancient DNA and modern population studies. Kurds show the strongest continuity with the Sumerians, sharing high levels of Zagros, Anatolian, and Caucasus ancestry. Persians are also genetically close, though with slightly less Zagros ancestry than Kurds. Iraqi Arabs have significantly more Levantine and African admixture, showing they are less genetically similar to the Sumerians. Now, finally, let's compare the DNA genetics of the Kurds and Turks, the Assyrians and the Sumerians, DNA results which were made from the remains of that period, and let's end our topic without further ado. Kurds are genetically closest to the Sumerians, sharing the Zagros-Anatolian Caucasus triad with minimal external admixture. Assyrians are similar, but have slightly more Levantine and African ancestry, reflecting ancient Semitic expansions. Turks have more Anatolian ancestry, but are heavily admixed with steppe and Central Asian, Turkic components, making them more distant from the Sumerians genetically. A diagram representation of the genetic relationships between ancient Sumerians and modern populations. Kurds and Sumerians are from the same ancient genetic family. While they are not the exact same people, modern Kurds are likely direct descendants of the same ancestral population that lived in and around Mesopotamia and the Zagros Mountains, which also gave rise to the Sumerians. To clarify, Sumerians were an early civilization that emerged in southern Mesopotamia around 4000 BCE. Kurds are genetically very close to the people who lived in the Zagros and northern Mesopotamia, including the ancestors of the Sumerians. This is why geneticists and historians often describe Kurds as the closest modern population to the Sumerians. Based on current ancient DNA studies and comparisons with modern populations, Kurds are among the closest living genetic relatives of the ancient Sumerians. Here's a summary of why. Genetic overlap. Ancient Sumerian DNA, taken from remains in southern Mesopotamia, shows significant ancestry from Zagros Neolithic farmers, Anatolian early farmers, Caucasus hunter-gatherers. Modern Kurdish populations show the highest continuity with this ancestral mix, especially with the Zagros Neolithic component, which is the genetic core of the Sumerians. They are likely the most direct genetic and cultural continuation of ancient northern Mesopotamian peoples, including those who helped shape the Sumerian world. Recent DNA research reveals that the Sumerians possess a strong genetic affinity with ancient Zagros populations, suggesting deep roots in the region. This genetic signature closely aligns with that of modern Kurdish groups, indicating that the Sumerians likely descended from a lineage connected to the ancient peoples of the Zagros Mountains. These findings support the idea that the Sumerians were an indigenous civilization, emerging from the heart of the ancient Near East with lasting cultural and genetic ties to the land.